This is Ross Boucek talking about climate extremes, create legacies, and tropical floodplain fish subsidies. Good luck.
Thanks for all sticking around. It's been some great talk so far. Been learning a lot. So forecasted changes in global climate predict not only shifts in average conditions, but also the increased frequency and intensity of climate extremes. Climate extremes are defined as discrete weather events that fall within the statistical tails of some climate parameter, and are often characterized by being relatively rare and relatively intense. These can include, but are not limited to, hurricanes or extreme wind events, extreme temperature events such as heat waves, and extreme precipitation events like droughts. Climate extremes often create conditions that exceed the acclimation capacity of many organisms living within the impacted regions, causing punctuated killing events to those species and essentially causing communities to reset. Though there's been some work examining within community effects of climate extremes, the very few studies have really examined the effects of climate extremes on the exchange of biomass from one community to the next, or how climate extremes influence subsidies. Upper trophic level subsidies, or subsidies that are essentially not a basal resource, may be particularly vulnerable to these disturbances as donor communities reset and recover from these extremes. For instance, take this couple trophic level donor community. It provides resources to this recipient community. The donor community is then impacted by a climate extreme, essentially causing the community to reset, decoupling the two communities. However, due to a slow recovery of your top predators and a subsequent predation release of the prey, you may expect a temporary increase in the subsidy to your recipient community before the trophic structure of the donor community fully recovers and the subsidy goes back to normal. The coastal rivers in the Everglades provide a good model system to test the effects of climate extremes on trophic subsidies. For instance, seasonal rainfall patterns largely dictate a loch-in prey availability to riverine consumers, or dictate subsidies to these consumers living in the river. In the wet season, vast rainfall increases, causing vast expanses of marshes that crown the upper part of these coastal rivers to become inundated. These harbor larger bodied donor community top predators, as well as spawn the growth of many donor community prey. In the dry season, these marshes dry, forcing fishes into the deepest habitats of the landscape, including coastal rivers. As these fishes enter their estuary, a common river predator, the common snook, triple in abundance, presumably to take advantage of these resources. However, how much resources available may largely depend on the population dynamics of your donor community top predators. Because the productivity of these donor communities is largely dictated by rainfall patterns and the inundation of these marshes, they may be particularly vulnerable to droughts. And in 2011, we had one of the worst droughts in 30 years impact South Florida. It's represented in this figure where you have the number of days marshes or donor community remained dry, index of droughts, time on the x-axis, and you can see the 2011 drought in red essentially caused marshes to dry for 100 days, tripling the average number of 30 days marshes are normally dry, providing us a unique opportunity to test how droughts may influence these trophic subsidies in these coastal rivers. So our research questions were, did the 2011 drought actually impact our donor communities? Did changes in these donor communities actually alter the magnitude of the composition of the subsidy going to our recipients' consumers? And did changes in the magnitude of these subsidies actually result in the changes in composition? To answer these questions, we conducted a 3-year study tracking the donor community top predator abundances and recipient community top predator abundances, the biomass fluxing into the rivers, and the consumption rates of our donor community and recipient community top predators. So our model system we chose is the oligochalane reaches of the Shark River, occurring in the southern Everglades. The Shark River connects the Shark River Slough, the largest freshwater drainage of the southern Everglades, with the Gulf of Mexico, and is a focal site of the Florida Coastal Everglades Long-Term Ecological Research Site. This is an aerial photograph of our study sites. The brown surface represents the marsh, or the habitat for our donor community. The river here is our coastal river, and the white dots represent our 5 fixed study sites. One thing to take note of this is there's a very high perimeter area to area ratio in our recipient habitat, often indicating that there's a high sensitivity of recipient communities to allotment resources or subsidies. In order to track the full dynamics of this subsidy and how it changed over following the drought, we conducted a 3-year study sampling these 5 fixed sites once a month, from essentially as close as we could get to the peak of the wet season in December, down to marsh floodplain drying, and back to the rewetting again in June. This is hydrographically reflecting that. On the y-axis, we have basically marsh stage. On the x-axis, time, and the gray bars represent our periods of study for each year. We sampled the abundances and biomass of fishes in these rivers using electrofishing methods, where essentially applied a current to the water for 100 meters of shoreline and netted everything that's immobilized. And we conducted this at each one of these five fixed sites with 3 replicates, and did this every month. We focused our analysis for our top predator abundances and how they may have changed following the drought on 3 common predators within the system. 2 being from the donor marsh community, this being the largemouth bass and the bowfin, and 1 being the recipient community, snook. The pie chart is essentially the percentages of each species out of our total top predator abundance. Gar represent 50% of it. However, due to their very low feeding rates and low dietary overlap with our recipient consumer, we expected their per capita and probably the population level impacts on the subsidy to be pretty minimal. And I hate to say this, but we compared annual abundances per 100 meters using paleo statistics and the repeated measures of NOVAs. But I'm excited to explore ways of doing some post hoc model selection analyses on these data. So, let's see Dr. Anderson. So moving on to how we track changes in the dynamics of the subsidy. We measured the amount of total, or the average biomass per 100 meters on a monthly basis from electrofishing. We then took advantage of our continuous monthly sampling to develop biomass curves. We then determined the yearly relative magnitude of each subsidy by calculating the area under these curves and comparing these areas across years to determine the relative impact of the drought. The subsidy can really be packaged in 2 different ways, same species but different size classes. You can have these large sunfishes or these kind of hamburger-sized meals, or it comes in this really numerically dominant, somewhat potato chip-sized subsidies. So we broke our analysis up broke our biomass curves up into these 2 different functional groups. We measured our consumer diets by essentially we removed consumer diets with pulse gas to clavage. It's non-lethal, pretty effective method of doing it. And like we did for the prey, we calculated the average biomass consumed of this floodplain subsidy per 100 meters of shoreline. And as we partitioned the biomass curves into 2 larger and smaller sunfish size classes, we partitioned the total biomass consumed by either whether it was diverted away from our donor to our recipient consumer by our donor community predators, or was it actually in fact going to our recipient consumer. And again, we calculated the area under these curves and compared them across the years with chi-squared tests. So moving on to hypotheses. On the top, you have the abundances of our top predators from our donor community. Pre-disturbance, we'd expect them to flux in as marshes dry and then leave when marshes become inundated. We expect marsh prey to follow the same example, but instead of leaving, they are getting consumed. And some of our previous research has shown that when this dynamic happens, the amount of biomass consumed is roughly 50-50 split between a recipient consumer that actually it's going to its desired or the location for the recipient consumer, or being diverted away by our donor community consumer. We expected, following the drought, we expected the marsh communities to essentially reset, resulting in no predators fluxing into the system, consequently no prey fluxing into the system, and a trophic decoupling between the marsh and the recipient consumers. However, 2 years after the drought, we expected a slow recovery rate of our top predators, resulting in a top-down release of predation on the marsh surface during the wet season, resulting in more subsidy fluxing into the system. And because there's no consumers following it in from the estuary, we expect most of it all to be going to snook. So it's temporarily increased subsidy to our recipient consumer. So moving on to data. On the y-axis is the number of fish per 100 meters. This is for our top predator abundances, and the x-axis is time. The green lines represent our donor community top predators. The blue lines is our recipient community consumer. And you can see they kind of come in, but we had a drought that happened really at the end of this. So as we expected, following the drought, you can see there's a substantial, significant decrease in all of our donor community top predators, indicating they really just didn't come into the system. They were, as a community, as we thought, were set. However, unlike what we expected, 2 years after the drought, we see a super resilient response from our top predator community, where their average abundances in 2013 doubled that of what it was in 2012 or 2011. So not the slow recovery, but instead overshooting what we thought the pre-disturbance numbers would be. So moving on to prey, on the y-axis is the average biomass of sunfishes that moved in per 100 meters of shoreline. How to interpret these figures is these top points represent the total biomass fluxing into the system. The white area under the curve represents how much of that biomass is tied up with our smaller potato chip-sized sunfishes. And the gray area represents how much of the biomass is really tied up with these larger hamburger-type sunfishes. And you can see in our pre-disturbance year, it's roughly 50-50 split to the numerically dominant sunfishes to the kind of rare, larger-bodied sunfishes. So we see following the drought, like we expected, no sunfishes flux into the system. Again, this probably community decoupling is essentially. However, in 2013, despite there being a doubling of our marsh donor community predators, we still see the biomass that fluxed into the system actually tripled that what it was pre-disturbance state. However, now the biomass is essentially all completely tied up with our larger bodied sunfishes. So the composition, we were right about the increase in magnitude of the subsidy, but the composition of it did change. So moving on to consumption rates. This is on the y-axis. This is the average marsh biomass consumed per 100 meters of shoreline. On the x-axis is time. How to interpret these figures. These, again, top points represents the total biomass consumed either by our consumers from the donor community or by our consumers from the recipient community. The blue shading under the curve represents how much of that biomass is consumed by our recipient consumer, and the green shading represents how much of that biomass is being consumed or being diverted away from our recipient consumers by our consumers from the donor community. So following the drought, we see, again, this trophic decoupling. No marsh biomass was consumed, and it's somewhat more exaggerated than what we saw when we measured just the average abundances of prey. However, following the drought in 2013, or 2 years after the drought in 2013, instead of seeing a high consumption rates that's matching what we were measuring in our abundances, we actually see the area under this curve does not differ from the area under this curve. In fact, the only real difference is now that the proportion of the biomass being consumed is even less for snook than it was in this pre-disturbance year. So despite there being more biomass fluxing into the system, less of it's getting consumed by our recipient consumer. So much different than what we expected. So moving on to our hypotheses, we were right. The communities were reset following the drought. We got that one. However, this top-down release and this larger subsidy going to our recipient consumer, we're a little off on. What we did see, what we saw instead was a super resilient donor community marsh, or donor community top predator community. We did see more biomass flux into the system, but it was tied up more on these larger sunfishes that may have been less exploitable than millions of these little numerically dominant sunfishes. And we saw no really changes in the consumption rates of our subsidy from our pre-disturbance year. However, less of it was really going to our targeted species or the recipient consumer. So though we're still trying to figure out the mechanism of the second year, one thing we can learn from this study is that as these climate extremes increase in frequency, we may expect the year-to-year predictability of these subsidies to decrease, which will undoubtedly have effects on the recipient communities. That, I'll take any questions. Julia, so kind of first.
First of all, next job, and I'm asking this because I know you like other extreme climatic events. So how are you decoupling that cold snap that happened, or the timeline of that with this stuff?
Well,
or are you putting it as another parameter?
So we're very lucky in our freshwater communities are largely temperate, not affected by cold fronts. So at least the donor community dynamics was not affected. But you can see, if you take a closer look at some of the abundance graphs, our recipient consumer is slowly increasing in abundance. Yes, Dr. Karnowski.
My thought would be you might want to quantify the extremes in terms of the climate extremes. So like how many standard deviations away from what average or what time frame.
Mm-hmm.
That might put that in perspective a little bit.
So the standard definition of a climate extreme is one that falls within the 1% statistical tail of the probability distribution, which this didn't. We have records and we have a paper that's recently accepted pending revisions in global change biology that's showing this was not in fact a true climate extreme, more of a climate episode. But it does, it was the worst drought we've had in 10 years and likely created conditions that was never experienced by this donor community. Yes, Zach.
Well, so, two questions. The reduced predator abundance that you saw the year after the drought, do you think that's mortality, or do you think they're utilizing different parts of the system? And then the second question is there any chance that increased salinity during the drought has reduced the effectiveness of the electrofishing, and that's partially explain the results you saw in reduced predator abundance?
Well, where we saw the reduced predator abundance was 1 year after the drought and the high salinities. So if I plotted salinity here, it was essentially fresh all year round. So your other question, whether or not this could be some sort of immigration response and then they coming back, is a possibility. But we have data from canals that would be the other analogous dry season refugia that show the same pattern, or the same pattern with their predators, where in the canals there were no predators really fluxing into the canals either in the dry season.
Fantastic. Thank you.